Okay, hello everyone. Welcome to today's TSVP talk. It's my great pleasure to introduce today's professor, Torbjörn Lundh from Chalmers University of Technology and the University of Gothenburg in Sweden. Professor Torbjörn has also served as Deputy Vice Chancellor at the University of Gothenburg, and he is also the president of the UNITECH International. He has also his research has evolved from pure mathematics to mathematical biology, with important contributions to evolution, artificial life, speciation, and population dynamics. So today, he will also talk about the process of evolution from quite different viewpoints. Like from the emergence of new species in common environments to the future coexistence between humans and AGIs. So it would be very interesting. So welcome, Professor Torbjörn. The floor is yours.
Thank you very much. I hope I put on my. Thank you. Thank you, Dr. Yang, for this warm, kind introduction. I especially like when you said my interest had evolved since this is a theme for today. It's a bit of Gothenburg humor, I hope. So I'm grateful to OIST for having me here, and also to TSVP for setting up with such a great leadership with Jonas here and Lynn that help us feel at home from the start. And it's really great to be here. And also thank you for coming here, so many of you, and maybe people from logging in. I don't know, but Lynn knows if there's someone out in the cyberspace. And maybe I will also have a premature thank you to those who will watch this later when I sign the form for YouTube. And hopefully even a stronger thank you to if somebody logs in at 2036. I'll come back to that later. The plan is that I will lightly pass through some evolutionary studies I've been myself involved with, just to show that it's such a broad area and such an important area. And the tools that I've been using is some sort of different versions of models. And I know that I picked that field because OIST is such a special place when it comes to evolution, with such great evolutionary people here, and also the possibility for collaboration between the different groups. And I will then try to do that kind of lightly, but please interrupt me to make clarifications, since I will move through this a bit, several different studies like that, and then to come to this possible future work and future concerns maybe about what will happen in the near future here. So nothing at all makes sense except in the light of evolution. So this is a homage of a title that maybe some of you recognize from another very important evolutionary researcher, Dobzhansky. And if we focus first on the words "make sense" here, what do we mean by that? This is the original title here that I tried to make an homage for from 1973. And Dobzhansky then, he said something that if you want to explain something or answer the question why, you'll kind of fall back into evolutionary comments eventually. And this is true for everything biology. This is the ground theory. That's where you have to work and to explain things. And this has also been taken up to social biology, for example, Edward O. Wilson, just to mention one name there. And to couple this with this wonderful environment we have at TSVP, my friend and neighbor here, and he's here, with his lovely fiancée or girlfriend, or not wife, for sure. Hannah is here as well, thank you. So you have a slide with the headline, why? And I think that was very suitable this week when you were standing here last week. And I will repeat that here. I'll steal that Will Horowitz slide. And but I just want to say that you're not the first one to ask the question. Already the old Greeks, as you say. Thales, he was supposedly the first one to put focus on that. That's when the modern science started. Instead of just giving facts or statements and predictions, he was asking the question why and trying to force people to answer that, himself included. And since then, these colors have been flying high through the academic society. We take pride in asking questions why and trying to answer them. So what tools do we have when it comes to why questions here? So I think that the one common big tool that is used all over the place are the scientific models. And since they're used by almost everyone, even those fields that really don't realize they are using models, like pedagogic, for example, where they're using theories, but they are really models. So they are commonly used, and they are used not in the same background. So that means that there could be some confusions when we do this sought after interdisciplinary works. So me and my colleague, we were looking into this. And maybe you recognize this famous quote by George Box, where I think it's kind of cool, that we want to be as stringent as possible, especially in mathematics. Exactness is everything there is. But then we talk about this fussiness, the useful fussiness. And the paradoxical thing that if we are trying to be complete and exact, we're dealing with theories. But the really useful things are the models. So we tried to write a book, or we wrote a book. We tried to overcome this culture difficulties sometimes when you have collaborations. I know that some of you might have had that experience, that you're talking about something and it seems all fine, but then it seems like you have misunderstood each other. And not too seldom is that about your viewpoints on models. Because in medicine, for example, the model could be C. elegans or a pig. But in the mathematics, it's maybe a PDE model or SDE or ODE, as we talked about yesterday, the chairman and I. So we tried to write this book as a culture overbridging book that I interviewed, for example, 10 researchers, and we were then with the same question, so the reader can see the difference in the culture here. Because then I tried to come up with a funny subtitle. I don't know if you find this funny. If I say red atoms, what do you think about then? What atoms do we talk about?
Soviet atoms.
Soviet atoms. But we got some chemists here, I think. Red atoms, which are they? Or are you shy? Or is it, maybe it's a Swedish? In Sweden, we had these red balls as being oxygen all the time. Yeah. So, but that does not make, they're not red in your domains. You don't have that light, right? So there is no color in that sense that we see. But we are used to the red is oxygen. And they have some holes. The oxygen, atom has some holes. You can poke some bars there to add the other color, right? So that's a model that's creeped into our minds very, very clearly. The white lies, of course, and black boxes. What about yellow book? For non-mathematician, this Verlag, Springer Verlag, they do all the math books in yellow. So that's it. So I've heard mathematicians speaking on the phone. Okay, I'll meet you on the platform. I will be the guy with the yellow book. So it's a way to recognize the other one. Okay, sorry for that diversion. But my dear collaborator and friend that he's been working with since he did his master thesis, Philip Gerle, most of the things I will talk about here is together with him. So it's really nice to have him. So I try to make him a visit here, but he is father of 2 youngsters and trying to make life work. But I'm really grateful with that collaboration here. Okay, so that was a little bit about making sense. What about the illumination focus here, the evolution itself? What do we mean, or how do we study that? So the main obstacle about study evolution is it takes such a long time, right? So it's hard to do experiments with that. People have been doing that. For example, Richard Lenz, he has set up a lab. It's been going on for over 30 years, maybe 35 years now, where he studied E. coli, tried to capture what's, the evolution of that. And that's a really time-consuming, of course, endeavor with a lot of energy to do that. But of course, we can also use these models now. So for example, we can also try to answer concrete questions about evolution. So I'll try to mark them in blue here, the questions. And we'll start with one here that is hopefully suitable since our chairman, Dr. Yang, is a sympatric speciation specialist. So I just want to explain the word sympatric speciation. Are we having a guest speaker? No. That sympatric means the same fatherhood. So that you are in the same space. And is it possible for organisms in the same space to start to divide and become two different species? That has been a long-standing question, and of course, many answers to that. But one big answer came with Ulf Dickman, the leader of your group, Yang, and also we have other members here today, and his collaborator, Michael Döbli, show that using a discrete logistic model and modeling that with agents to come up with the origin of species. And here you can view this now as you have an ecological character, that meaning you can think about that the beak size of a bird, and here is one single size, and then there are two food sources. That means the grains with different size that fits different beaks. And you see that this can now be endeavored by using, in this case, that you also have a preference. You can become yellow, and you can also evolve an attraction to yellowness. So therefore that you can sweep out the hybrids in between by doing that. So this is a tool using ordinary differential equations and even also PDE if you want to add space there to show that, okay, this is feasible if we accept this model here. And we did something back in Chalmers, some things we tried to repeat more or less that. Actually, we didn't know about the first of Ulf's study when we started, but we did it differently from using statistical mechanics, kinetics, and also added sex to the equation. That's always complicated. And we got some similar situations here. And here there are different food sources, and here we talk about the attraction and the expression of different maybe colors.
What is the second modification?
Oh, yeah. Nice, thank you. So this is a situation when you have three food sources. So these are two independent runs, and you see in the first case it became a one size fits all bird here that could take sources from in between. But then he made a second split, and in this one there was some chaotic behavior here, and they got a triple all around. And up here they have that you can, independent of that, you have something that enforces that you can see before that. Okay, I really want to go for this yellow lady over there, or it's probably the other way around. The man or the male is a yellow bird, and because there's always the female that chooses. Thank you for asking. Perfect. And okay, so that was the situation we feel most of us not really comfortable with. I think we're using equations like models like that. But I just want to push a little bit for another way to do research, which is fun because that's about playing. And this is a book called The Playing Man or Human. And this is a way to understand that we all did when we were kids. We played around and we got a feeling for stuff. And I think that's really interesting when it comes to complex things such as evolution. So I will come into like a way of studying evolution in a more playful manner, I think. But you can always, of course, it sounded like unserious, but it's still serious play. Play is maybe always serious in one sense. And look at the artists, they just. So we have then a way that mathematicians and physicists always do when we come into a problematic case. We try to make it more general and more abstract to simplify. It sounds a bit paradoxical, but by doing that, you can maybe see some paths that are hidden when you look at all details. And if you look at evolution as a process, what kind of ingredients, what is the big thing that you really need to have, and that are the essential part of evolution. And this is the trinity. You need to have inheritance. You bring down to the new generations the information. And then you will have also inheritance with some errors, which we call mutations. Because otherwise, nothing new will happen. Just send down a copy. And also, I mean, this sending down information is also a hard thing. So it looks like an after construction because it always can be wrong sometimes. And the third thing in this trinity would be then competition. So you need that there is a competition between, so not everyone will send off the same amount of offsprings. But if you have a competition, you will have those who will be best in some fitness sense. They will then have more offsprings. That trait will spread around in the population. So the idea here is to set this free by constructing a model in the computer and not aiming for copying the carbon-based life. Just looking at this thing, can we play around with these kind of basic rules and see what happens? And then can we find some universality that we can take back as lessons to the carbon life? So go back to a little bit the picture from the book here. We try to look at different types of models in this modeling house. We have the conceptual basement where we don't have any quantitatives. We have only arrows, and this is affecting this, and there's a feedback here, but we don't have any numbers. And then we have an analogous billiard room. So we can have a pool table here where we represent the universe, or we can think about that, the other, what would we say, the poster example would be that the Bohr's atom model was an analogy of the planet motions around the sun, right? So this is comparing with something else that we know more about. So what I'm talking about is doing this analogous thing and maybe conceptual, and that's a really bad resolution. So my first PhD student would be very angry with me if I showed this bad resolution. Sorry, Jonathan. Anyways, so let's move on to what I'm talking about here. The artificial life is a field that is studying evolution as a process without caring about the outcome, just to see what happens, and by setting up these rules. So the player is to try to make this as good as possible, and have a bit unsurprising results, but with as few and simple rules extra except for these three as possible. So the one that I will talk about the result is when we did studies with a special artificial life program called AVIDA. That was a successor of another Spanish-sounded program called Tierra. So AVIDA is then a computer system, or there's CPUs spread all over the place. So there's a lattice with all these CPUs, and every CPUs have a memory, like a band here that goes around. And the whole thing is to try to copy itself and place it to a neighbor. That's fine, Edith. Sorry, I shouldn't name you.
Tell you later the reason.
Okay. So this is then the program that runs and tries to copy itself and place it to a neighbor, randomly shows a neighbor. So these, as you can see, that maybe if you recognize that from, it's very ASCII-like things, because the program language usually is a very, very brittle thing. If you make a mutation in a computer program, usually everything crashes. And that is very unbiological in the sense. So usually we think about DNA as being a program, but that part is a really bad analog because the DNA is super robust. So we need to have robust languages. We have, for example, no operation. I don't see anyone here, but no operation. Just a command, an empty command like a junk code. And you got also this So you have a robust that can withstand point mutations. And then you have inputs and output in stacks. So this is like a side effect here. So if you are able to do X and Y here, and you do the AND logical symbol, so you see you have a 1 in the first place, and if that turns out to be Z here, on these registers, then you have successfully made an AND operator. So this is like a side thing, instead of copying itself, is also do these randomly operations on these stacks. And if you get a logical symbol, you get more time in the time slicing. You get faster. You get higher fitness. And therefore, you can get more offspring. So this is forcing the program to create more and more logical symbols, a more advanced one. The most advanced logical symbol is the XOR, the OR, but with an exclusive OR thing, right? Okay, was that okay? Very vague, as I said, light-handed. But it's a way to, that we have inheritance, we have mutations, but we also have competition with these logical symbols that you can create. And then you have a lattice of all these points are CPUs, and these are the same, they have the same genome. And so you can spread with different colors here and see what color will take over. And then will come another one because they learn to do something more. So it's an open-ended system like that. And the thing is not, of course, to create this logical symbol. It's just to see how is the action going on with it. And so that has been a nice playing around. And we use that also in teaching. There's like an education version of this. You can have this agar plates and play around with it. It's kind of fun. I think you get a feel for it. Just to make a quotation here, that was from 2005 with the Richard Lenski I mentioned, with the man with the lab. 30-year-old lab. He got a copy of this, so the creators from Caltech sent that to him, and he played around with it, and he was tempted to close down his laboratory, because he got a feel for evolution or smell, as said there, just by playing around with it. So I think that of course he's fighting a lot. Luckily, he didn't close his lab, but it gives you another flavor. I think it also pinpoints the way that it's very good to be able to use real experiments together with models, and not only maybe one model, but maybe you can have a PDE model and this kind of agent-based model. And the more different viewpoints you have on a complex phenomena, the better picture you might get. Sorry. Is that okay so far? Great. Such a good audience. Okay, so here's another question. Is gene-to-gene networks, so that is, gene-to-gene network is genes that will be connected in a network if they are occurring in the same pathway. So pathways are the processes within the cell, and they are building on different proteins, which are then coded in the genes. So if the same genes show up in the same pathway, they are connected with the edge. So then you got a gene-to-gene networks. And there was, at that time when we did this, there was a debate in the literature between different groups saying that they were seeing some kind of, this is a typical biological scale-free network that shows up all the time. Or no, this is exponential, said another fraction. And what I mean by that is that you look at, let me do that here. You look at a random node, and you see what is the probability that that node has a degree, that means outgoing edges, that is higher than k. If that scales, or is estimated by constant times this. If that scales looks like that, e raised, they call it exponential. We have the exponential here, negative. So usually have this function, it's very many with just one, and then it goes down like that. But does it go down maybe in this way instead? This is what they call a scale-freeness, because it looks the same. It's like this fractal structure. It looks the same on different levels. So that was a bit of controversy. We thought maybe you can use this artificial life, the Avida here, to look at this. So that's what we did. But we started by looking first on the way that this has been formulated. This is the gene-to-gene network, because they are connected if they are in the same pathway. So pathways are the squares up here, and the genes are down here in the circles. So you see, if you put this together, you will have the same gene-to-gene network, but this is slightly different. You see this edge here, for example. So it doesn't give the whole information. So if you keep this structure instead, you have a better information. And you have this is called not bipartite graph, because you have different nodes downstairs and upstairs. And you can still talk about what is, has degree 3, this is 1, this is 2, this is 2. So you can still talk about the degree on the gene. So you can ask the same questions. And that's what we did here with the yeast. I will blow up here. So we have pathways here, 120, and a lot of more genes, 450. And you put that up here, you got the network, bipartite network that looks like this. And if we now look at the degree here for the pathways, which we can say is the function F here, PF, you do this log of that axis, the probability, and K here, you get more or less a straight line, which tells us, oh, this is now a scale-free network. And if you do the same thing with log-log, then you capture the exponential part. And that's, as you can see in this one, looks like a straight line. So that means that we can answer this question with a yes. Right? So both groups are right in some sense. So if you mix them together, you have both at some level. And okay, I said I was trying to do this also with the comparison here. Here's the blow up of this previous one. And this is now the Avida one. So did I show that? So you see, this is what we saw before. And now this is what we had with the Avida one, where we have the genes being the program lines, the codes or the commands, and pathway, the function, the logical functions that you needed to. Now, I should also confess and feel it for forgiveness, like the biathlon runner. So sorry, feel it, because this all started when you asked for doing a master thesis project with me. And I heard before that the creators of the Avida, they said that the evolving program that is being created is totally unreadable. No one can read it because it jumps back and forth. It's super bad programming. And I gave him that problem. You have to learn how to read this code. So at some point, he was the only one who could read it. And that was really fulfilling, because then we can start to see what's happening. And it's also a strong analog what you see when you look at the biological DNA. It's really hard to tell the difference just from the outside, which is coding and non-coding. People thought for a long time you can look at the entropy, the order, and see that if it's really a coding part, then the order must be kept. But that part looks as messy as the random junk code. So I think that was a bit revealing, that we can find something that is universally like this. It doesn't really work low and really not high, but it's some part. So of course, there's room for improvement here. That's still okay. So that was one answer. Was that okay? This is a leading question. Can I move to the next one because we're running out of time? No. Do you want to ask more about that kind of study? Yes, thank you.
A quick question. Overall, do you think whatever structural feature you observed resulted from some kind of selection for that certain pattern, or it's just a spontaneous pattern that emerged in the absence of selection?
Yeah. Okay, so can I don't know, can I reformulate the question? Is, do we trust this as a uniformity that you have, that you can transfer the knowledge on this computer program to the DNA? Is that
no? I'm just asking about the interpretation. What's your interpretation of your observation?
Oh, okay. So the one that from what I've said to you, that you can, I would say that this playing around with this in silico model, this artificial light programming computer, can give you insights, because in this sense, the output was a bit similar when you talk about the output as being the degree distribution of the gene to phenotype bipartite graph. But then we did dig a bit deeper in that and looked for how did a new function come to be birth, to come to be created here. I had to avoid the word creation, but let's use that anyways. So how do you get a new function? And it turns out that nothing is created from scratch. It's always you have to copy some parts and put that together. And if you are, since I know that many of you have been following fossil records and things like that, that it's really hard to do in real life. So that would give you a way to study this in action, the evolution in action. So that is my interpretation. That.
Oh, sorry. I guess my question was clear enough.
Yes.
Well, what I mean is, say, I know that in this artificial life system, there exists a selection for the ability to replicate.
Yeah.
But what I'm wondering is, so does these patterns in the gene network indeed related to their ability to replicate, or is it some emergent pattern that would emerge no matter how you evolve them?
Oh yeah. It's very important to have that competition. I agree. And you can, in this artificial life program, you can change the evaluation system, and you can play around with that to feel the robustness of that as well. And you can also have, there is like a difficulty to reach the XOR, for example. If you don't give any real good reward on the way up, that will never happen. These higher orders of difficulties. Was that? Sorry if I don't misunderstood it again.
Yeah, thank you.
Okay. So it's a very essential part. And so if you turn off that reward, everything stops.
Thank you.
Thank you. And I will also say that eventually, when you're done with these logical operations, there are no more rewards. Then everything also comes to a close. So let's jump into the next evolutionary question here. Okay, so remember the first one. We had sympatric speciation, where we had different food sources, different grain sizes, and different beaks. Is it really necessary to have different food sources, or can we actually, in sympatric situation, have speciation just with one food source? So that's one question. The other one here, if you have a system, ecological system, and you add more energy to it, maybe more brain nutrients, for example, what will happen with the diversity, the species diversity? That was also different schools talking about that. Third one, what are the necessary conditions, or can we have any conditions where we can have coexistence with different species? The problem with evolutionary theory is like, not a problem, but one of the things that it tells you that this fittest of the survival, and that is only one person or one species in this mixture. If it's just a one kind of area with one kind of food, in that sense, that's easy to think about. But actually, can you still have something here? So this was inspired by an experiment from Helling and his group, but it's also an important question itself, because we know that the most of the cells inside us is not our cells, but actually in the gut. These vastly outnumber the human cells. So, and you also know that this is something to do with how wellness ourselves, if we have a good ecology with the bacteria inside us. So it's important, the coexistence here. For example, if you have taken if you've taken penicillin, very strong, broad range, then you don't feel as good. So that you need to build that up again. So this experiment was that they had a chemostat, Helling and his collaborators, and they had sugar in with one type of sugar. They had one strain of E. coli here, and they had a mixture, and then they took off the same amount that they added, like waste products, and they kept that as like a steady state. And to their surprise, I will see here, this is from 87. To their surprise, they started with a single clone. After 765 generations, two majority and 2 minority types took clones that were then identified. So it could even be more of these E. coli in that system. So that's typically speciation with one food source in a well-mixed sympatric environment. So it's a bit of a mystery. But it turned out that the reason was that the bacteria that took the sugar didn't take all the energy from that sugar part, but actually left over is a way to say it. The leftover was then in the mixture, and that created a secondary food source, right? The pre-chewed sugar canes that maybe was better specialized by another one that mutated and took over that. You see the point? So it's like one food source in, but it was then remodeled by taking out some energy, but not everything. So by living on this leftover, that's called the cross-feeding. Okay, so we try to do, can we do a model of this one? And we have a strategy, Philip and I, that we try to start usually with agent-based things, because they are easy to grasp, and so you can get some statistical result, and then maybe you can also do a model in the more proper, but the old school way with paper and pen, and do an analytic version of that. In the same philosophy as more model for the same kind of phenomena. In this situation, we wanted to make a model like an artificial life model, which means that it should be free and just roaming around. We didn't care about trying to be, mimic the nature of E. coli or anything like that. And we wanted to build it with as simple Lego blocks as possible, well-known, hopefully. So see if we succeeded with that here. So this is a startup here. We have food coming into the system, which are binary strings, zeros and ones, and with a special inflow rate, gamma here. And then these resources, Ri is a piece of food, binary string, and then these agents, AJ, AK, for example, they are eating that, chewing on that one, and then it will change to another food source, R prime, and you can measure the entropy difference. And the entropy is a measure of orderliness. If you look at what Schrödinger said in his book, What is Life? When he wrote that in Dublin, he said that life is something that sucks orderliness out of its environment to keep its integrity. So here, if you can suck orderliness by increasing the entropy, the amount of unorderliness, then you're taking energy, and then you can be rewarded with some probability depending on how much energy you take out with an offspring of your same sort. Except it could be a mutation, so it could result in another sort. Okay? And then this waste or leftover is taken up with another agent, AK. And if that one is able to do that, it will repeat here. Do you get that basic thing? So thank you.
Because it's a binary string, you're going to be having the maximum entropy that you're going to be having is like something like 1, and it's going to go down or up somewhere between 0 and 1, right?
Yeah.
And then after a certain level of consumption of that value, then how you're going to be keeping track of any change?
Yeah, then it's you have taken out the energy. Yeah. So but you have in the system, you have inflow, but you also have the outflow with the same. So you have fresh strings coming in all the time.
Oh,
otherwise everything would stop. Thanks for picking
that from the starting. So it's not like some initial condition from then on you're going to be considering, but every here and then you're going to be adding.
Yeah, exactly. So you have this system like you have in the living thing, right? So you keep that at some energy input and taking out that. Okay, good. So you pick random strings to take out. Yes, thank you.
So does it mean that the probability of mutation or something depending on the difference of entropy in the system, the P delta is something. Does it mean that the probability of having offspring depending on the
yes, yeah. If you're able to suck a lot of energy out from it, you have a higher probability to have an offspring. If it's negative, you can chew it and it can increase, right? in theory. Then you will never have an offspring. So, yeah. Is that okay?
Yeah.
And as agents, we will try to pick something that we all know. That's basic fundamental cell automatas. These one-dimensional ones that Stephen Wolfram, counted from 1 to 256. These guys, right? These rules that you see. This is rule number 30. So you can view this as a binary when you have black, 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 or 1, 1, 1, up to 0, 0, 0 at the end. So, and then you see that you have 4 ones in the middle there. And then if you sum up that in binary, this is 2 up to 1, 2 to 2, 2, 3, from 4, and you add that together, you get 30. And then you see that this is the rule 30. And if you stop with just 1 here, 1 black in the middle, which is really, really low entropy, then this will increase. Yeah. And 30 is like one of these magical in between chaos and orderliness. It's okay. Is that okay? Yeah.
Sorry.
Thank you.
Regarding this problem about speciation in general, I just want to confirm what's your operational definition for speciation here? Is that different from your operational definition when you deal with species doing sexual reproductions?
Yeah. So here we don't have any sex at all. And we have phenotype equals the genotype. So all the organism is a rule. So if you have a mutation, you jump to another rule with a certain problem.
So does this mean that you are not going to model the mixture between lineages?
No.
So
exactly.
Okay, so then once they are different, then they are different forever.
Yeah. Well, until they will die here, right? So you have death as well. So you have the same amount of members. Yeah. So when someone is born, someone else is taken out of the system.
Yeah. Thanks.
Yeah. Thank you for clarifying that. And it's a big weakness that we don't have a genome, because if you have a genome, you have another level of interaction beneath the surface that can create so much more information or so much more surprising evolution. Here we already know these are the species that we got, but it's computationally very heavy to do that. So that's why we moved from artificial life to artificial ecology, and then we gave up that kind of genome. Thanks for that. Okay. And then we could put together all this in a study. You have total 56, you start with that, and there is one that is really like this. But sometimes this one takes over and it goes back and forth and it never stops. It's kind of fascinating. It never stops. There will be new ones. And this is the mix of the food strings. You see that kind of dark. So that means that there are a lot of fresh ones in the mixture. So you can also change the flow rate here by adding more or less. And then the mutation rate, of course. And then you can see what kind of, who is eating the leftovers from food. So you get this food web things here. And then you can look also the whole population and see what is the reproduction rate here. How often do you get an offspring? And you can also look at the energy, how much energy of the strings. You can look at the energy when you put in and then they take out. What is the difference here? How well is the population taking out all the energy in the system that is given to it? So it's kind of fun to play around. You can do that in real time if you want to do it. And you can answer the first question. If you have a higher flow rate, that means more energy into the system, right? More high energy binary strings. Then you see that the Shannon index, which is a measure of the diversity in the population, the number of species here, more or less, that this goes down to a minimum, and then it stays there when you have a flow rate, which means that at that flow rate, it's a high probability the first string you get is a fresh one all the time. But if you lower that, it will be more and more complicated interactions by second and third order when you get a string eventually to chew on. So that is one way to do it. It's not so surprising. You can look at this food web interaction, see exactly what is going on here. And I like this thing when you can do pairwise competition. You can look at maybe 2 species here, did I show that here? I hope I did that right. Yeah, this one here. So you have two different species. You can see the image up there. You have this 129, which you recognize as Sierpinski gasket, which is interesting in itself. If they are competing, so you don't have any mutation outside these two, just between these two. If they are competing, then you will see that there will be more or less of a steady state with a relation with much more of these 145ers. They are more able to take the energy in the system to extract that. So you get the level, and then you can view that in this matrix 149 here, for example. That is very white because it's so good comparing to others. You have this pairwise condition, and you can look more at more complicated food webs by putting all this together. And now when you get cocky, and okay, what about three? Can you have three species in a collaboration and coexisting? Will just one take over? So intransitiveness, rock, paper, scissors. Was that right? Thank you, Alex. So this is from Las Vegas and the World Championship in that. But you have intransitiveness, that one is beating one, then you might have a coexistence here. And we did a study of that using the same system, and we came up with these intransitive situations where you can look at it. We have three species with the axis, and we can look at the simplex as one, so you sum up to one here. So it's a two-dimensional picture, and you see this is converging to a point inside here. It could be also on the boundary. So there are some different ways that this can happen. This is where we're the intransitive ones. Okay, and you also have sometimes also some wandering system here. And here's a nice group I like, because we're looking for the pairwise friends, that these two have a joint coexistence. You see here on the side, if you start on the side, you will find a place here. What will happen if you have three of them together? Because they're all friends pairwise. Think about a group of three friends. I'm looking at you now. You can easily get together pairwise, but can you all do three at the same time? It's not evident, but usually it works. So by looking at that, we can get a table here that see if we do just a random collection of these of three species, we get a single corner. It's just one taking over. It's 72% here, and otherwise, you can add pairwise, that would be on one side, and a triple that would be inside. It's kind of unlikely this here. But if you keep yourself to this intransitive, this will be even more unlikely, which is a surprise. But if you keep with the friends, then you see it's overwhelming. Pairwise friends makes good triples. Good to know. Okay. And then we also, sometimes you just state questions. Sometimes if you do the research, you will find, oh, this is also an interesting question. Why didn't we ask ourselves that? And the question here was, the final population that it converges to, will that always be the optimal with respect to the social gathering of energy or not? In this situation, it didn't, but here it did. So how can you set up some conditions when that would be right? Okay, I will just, so what I mean here is, how can you set up pairwise interactions with energies transferring and so on, so you know that for certain we will have that they will converge to a maximum for energy uptake. So that we were able to do by using some Taylor explanation in this more old-fashioned way. So now I've been talking quite a lot about
this does not include any competition between the species, right? This is one single species.
Yeah. So absolutely. So it's the same thing as before. You get offspring, and then someone else is taken out of the system. So you have the same number of players. So that means that if you're able to extract energy, you get more offspring. So that's how you compete here. Yeah.
That becomes the stable fixed point.
Yeah. Okay, good. So now I've been talking quite a bit about evolution, and over this process, I've been so amazed by evolution, and it's also very unpredictable and innovative. So I have also noticed another thing that when it comes to modeling here, that has been a bit interesting. I said that before, that the flying high, the colors of asking the question why, But we also have in this symbolic case where I usually work in the mathematics here, because mathematics is the only field where you don't use models, pure mathematics. And that's paradoxical, because you need pure mathematics to construct new models, but using them in pure mathematics, you don't. That's the only one. This is a bit funny, but you still want to be able to do it. So they are the ones that make the new models, but the real people that understand the environment, everything, they are the ones that apply them in a good manner. And here we got the phenomenological corner. Yesterday we had a TSVP talk. There was a lot about phenomenological models there as well. And here's a black box, and it's halfway outside here because it's not really a proper model in our view, since if you look at this one here, the diagram that we had also in the book here, they have predictive power on one axis, and we have comprehensibility. How well does the model explain something? How well do you understand it? Will you get, answering the question why again here by using that axis? So when we talk about models before, maybe 8 years ago, we talked about this model makes us understand something. But today it's more and more about predictive power. And this is, of course, of the large language models and machine learning that we have such a great success in making predictions. How a protein will be folding, for example. But do we really understand that more? So I think that it's worth to talk about this other axis here as well. Okay, good. So why I think that if we are looking at this flying high, the colors here, I think that I'm not sure if we really care so much about this understanding as we should in science anymore. It's more about making predictions now, due to the fantastic success of these AI models. So I collected a few examples there, but I really would like you to ask questions instead. So I just say one thing about the high. Stanford last 2 years ago, when the humanities AI group there had a guest from Oxford, a young, brilliant researcher who was really engaged and talked about her niece, that she had gotten a really good new question to ask herself all the time in the research, because the niece, 5 years old, running around and asking, why? And she's applying that to her research now. And I thought it was strange that not her advisor did that. So I mean, it's a super good question, but it's not the main core anymore, which is a bit strange, I think. So I'll skip the other examples. And I'll just say that in the October here, to 24th, so I had the honor to be in the panel with the group here from OIST, and we talked also about AI. And I remember I said, I have a really great talk, because yesterday Geoffrey Hinton got to be announced as the winner of the Nobel Prize in Physics, and he would hopefully bring up the existential issues about the future of AI to the table, and which he has really done. So I think that is really interesting. But on the same time, the evolution of that process has gone so fast. It's really exponentially how well AI can solve problems now. So it grows so quickly. And that is, people are talking about P-dome, what's the probability that AI will take over completely? And I don't know if you heard about the Faust parameter, which is like the next thing. And if you go around in Stanford or in Palo Alto, in these cocktail parties, as Alexander and I did when my wife and your sister left. So we had a lot of finger food at that time. And they talked about, what's your P-dome? It's kind of a thing that people discuss. And the Faust parameter is, what kind of P-dome do you accept, but still go ahead and do the AI transition to AGI? So Faust, the story about that you sell your soul to know everything, which is how much is that word? What the risk worth taking here? So I think that, this is not science. It's more of a social thing that we're doing here. Because if you look at with this boomer, what they call versus doomers, ethics also, people concerned about the security, it's also divided into groups, ethics versus existential concerns. And that's a friend of mine, Olle Häggström, talked about that a couple of days ago. He said that ethics is, right now, the AI is not good enough because it can give you biased results. And the existential concern is, very soon, AI will be too good. So therefore, there is like a concern about that at the moment. So, or I would say it's also like ethics that you can say that, can we trust AI? And the existential concerns is more, in a few years, will AI trust us? So this has been a lot of discussions, and there's also been what I heard a lot of comments like, oh, this is not science, so I don't care about this. But I think it's a bit sad, because we are in the university, we are not only doing research and education, but we also are doing innovation for the good of the society, but also serve the society. It could be not only innovation, but actually reading something, doing something, take part in the debate. And I think that is what's going to happen here. And it's even worse like that, because the internal value of doing research is much higher than education, and it's much higher than innovation. And at the end, they have served society when it comes to prestige. Here, this is a question about survival of the humans. So we need to take that, I think, seriously. And it's also kind of fun. So I would like to say that OIST would be the perfect place, having the strong evolutionary background. And I would love to, there would be a group here talking about using all that evolutionary knowledge, because we have people here that are experts in the rise of the phyla and the extinction of a group of species, for example. So just by studying, maybe OIST EGI would be a good name. And this might be a good book here to read. And just to take a quote here, that we need to use the best mathematical talent to address this question. And we cannot wait until it's on the doorstep, because then the AGI might even know what we're doing and try to shut it off. That's been already experiments in doing that. So I think this would be so nice if you could have doing something here. And I bought 4 copies of this book. This is the sharpest critique about the race to AGI at the moment. And even the CEOs, explains that this is the case, but the power of the market is so high that if you get it, you will become the king of the universe for a while. So if you want to join this, and I think it's a win-win thing, because if we are trying hard to come up with a baby step towards alignment and cannot do it, then that's also a strong signal that even people from OIST. So we should really put more money into studying this. Thank you very much for this. And I would just like to end with 2 haikus as a homage to the great island we are at. To play with fire, humankind got burnt but learned. Will we learn again? And here's another one. The child we create, we should coexist with it, else nothing makes sense. Arigatou gozaimasu.
Hey, thank you very much for your wonderful talk. So time for questions. Is anybody have some questions or comments?
Yeah, we have any comments.
Towards the end, when
Maybe you can go with the mic.
you were closing your wonderful talk, you started talking about this existential and ethical aspect, but these are really tools. Please forgive me, I'm asking your opinion. A tool itself cannot be an existential or whatever problem. Like, it is just like if you're going to be asking the question of because they don't have a drive.
I see what you mean.
Like an AI, it doesn't have any drive. It doesn't have any necessity that it requires to sell. The reason that the life runs is because at some point it's been said for whatever reason. There is no reason really. It's just conditions, constraint came together and the thing started moving forward. For AI, how would you really consider any particular AI-related circumstance that it's going to make the system, the tool, to run on its own for its own purpose?
So the famous example, which has been talked about so much, is by Nick Bostrom. That he talked about this paperclip apocalypse, right? So then you just tell an AGI to make as much paper clips as you can, and then it starts to do that. And that could be really bad, because also what Tchaikovsky said in this, before, that the AI will not be against you, but it will not be for you either. You consist of atoms that it might find some better use for. And then there is a drive, even for this one that I showed you. There is, because can you extract energy, for example, or can you do these logical operators? You hardcode that into the system from the start. So that's the whole point. Can we hardcode that they will still care of us? So one hopeful comment from Jeffrey Hinton the other week was, he said that in biology, there has not been one example of a more clever organism being run by a lower intelligent species, even though they are higher muscle mass or anything like that. It's always been that situation, except one case, which I think is kind of lovely. The mother of a newborn baby, the baby runs the show, and the mother is just, giving the baby what she wants all the time. So if we can hard code in this motherliness, love for humans, that will be something. But I don't know if it's a related. Was that a bit of an answer?
Absolutely. Yeah. Absolutely. Because literally, this was just the question.
Yeah.
I was not expecting the answer from it, but I was just seeing, what you look at, how you look at the problem, when you're closing this talk, how we are going to be actually bringing this whole together.
And it's, I feel like I'm really, against AI when I talk about this. You don't answer the question, but I really love it as well, because I started in the 90s looking at these neural networks, then as a model for how the brain works. I thought it's fantastic. You can use it like that way. But then again, I'm so impressed by, the evolution and following what's doing. It's hard to predict. It's so clever and come up with solutions you cannot foresee, which make a high, that I really have great respect for that force. But I really want to work more with AI. So I've written one paper in AI, and that is just because I was not taken seriously by the AI community at all if you don't write something. So I did that with one paper. And but that's, if it's, I would love to do that more, but I don't have the, what do you say, I don't know, it doesn't matter. So I would love to be convinced. I would love some of you smart persons would read this and tell me what is so wrong. I read it with that kind of viewpoint. I would like to be convinced and be carefree and work with AI, but I'm not at the moment. Will.
Yeah, so maybe provocative in that talk about evolution and sort of displacing one species with a new species. Would it be so bad if we created AGI and it wiped us out and then continued on?
Yeah.
That would just be our legacy in the universe, right?
Yeah. No. So
it's too much credit. Legacy, there is something going on. But legacy, there's no, we're not that special. It's just a life. And some constraint, some condition was met. If it was a little bit different, we wouldn't have been here, but some other animal might have. So giving up ourselves some credit of a legacy, I don't think we deserve that in this system. There's absolutely no value in us. It's just happened.
I completely disagree. The whole point of what we do is to leave a legacy to the world.
No, absolutely. We try to understand it, but
no, it's not about understanding. It's about what gives value and meaning to an individual's life.
There is no such thing.
Okay, I just fundamentally disagree.
No, but I'm maybe this sounded like a very provocative and strange question from Will here, which is my dear friend, but it's surprisingly many who exactly reasons like that today. The fraction of people say, let's go, we have done we destroyed the world anyways, we climate change and everything. And for example, Nick Bostrom, he has recently made a new paper that is let's download our DNA or a brain somewhere and just let it spread. It's because there are so many good things. We can cure cancer probably. There are so many good things that can happen. But I think we should talk about it and we should work with it, not just let some few CEOs in Silicon Valley decide the future. Sorry, Chairman.
Yeah, if they're still simultaneously emailing you, but about something else. So since you're a mathematician and this is a general talk, you suppressed the math, but maybe you can still indicate what sort of mathematical techniques or disciplines you use in your work on evolution. That's simulation and computer experiments.
Oh, you mean for the evolution?
The math.
Yeah.
Graph theory, is it game theory?
Oh, yeah. It's all of the above. So we have I didn't have time to talk about this game theory part, but there is a really interesting game by John Maynard Smith, when the animals are not killing each other, or the males, they're just standing there staring at each other and waiting until the first one gives up. Then he takes a better patch, and the loser will get this worse patch with respect to females or something. And the question is, how long should you wait? Because both are losing time. They're losing money by spending time here on waiting. So this is called the waiting game. So that is something that we worked also about that. So I mean, evolution is so complex. You need that game theory. You need this agent-based. You need EDs and SDs, because it's always this kind of. So that part is. But when it comes to AI here, we don't know. I mean, people have been saying, we should work on this alignment for 10, 15 years. There's not been really any steps for it. So, and for that case, I think we should turn around this square and look for service society. What can we bring here? We should give up that thing of being scientifically sound. How can we be helpful to society? And use our tools that we have, but instead of the goal of making papers, but actually saving humankind, which is nice.
Okay, Toby, thank you so much for this great talk, and also
you will explain why you're called.
Yeah, I will. Well, I have this call going on because we always buy granola at Wednesday afternoons. We can run to this guy. So I will give you one box of granola. It's magazine. But thank you for your lovely talk. Also, the way you connect with people and all the time ask, okay, is this clear? Really lovely how you do this. But from artistic point of view, I really love your visuals. So maybe you can send me your presentation, but also how important are these visuals for you in your work?
Oh, that's a good thing. So we talk about being a mathematician, and we had a discussion at the lunch table. And what is a circle for you? And somebody said, it's r squared equals x squared plus y squared. And someone, no, this is the geometry, the picture, the Platonic, ideal circle that goes on forever in resolution. And so I think you can divide us in different groups there. And I like areas where you can actually make a drawing. If you work in, for example, in string theory or in real abstract sense, it's hard to make drawings. I love drawings, so that's. I take this drawing class. So I think it's a personal thing. Yeah. That's yeah.
Great, thank you.
And there, if you want to join the book club, the Evolutionary General Intelligence at OIST group.